Today we will solve that 43 variant, May June 43, 2017. I gave you that past paper link yesterday. I hope you have that with you. So I'm sharing the screen. Someone is giving message. Okay, so I'll share the screen and you will get the past paper. Let's see. Now, this is our today's variant. Need more participants are okay. Muntasir came. If you say, Muntasir came. So, boys, 43 May June 2017. Everybody, go to page number 2, question number 1. And I want to take the test also, boys. So that test, I'll give you the question paper here. That first, 10 minutes test, not so big test, 10 to 20 minutes, like 20 to 30 multiple choice will be there. And then within a few minutes, like 2-3 minutes, you have to submit the answer. And I'll give you like 20 questions and you'll get 18 minutes like that, or 20 minutes maximum. And then you have to close your personal chatting box and all. Let's see. So get ready next week. Maybe I'll take a test, and you have to submit within 2-3 minutes. Now look at this diagram. Question number 1. This is the figure. This diagram in figure 1.1. There is a diagram of human heart. The diagram shows the phases during the heartbeat when the atria contract. Boys, you need to think about the cardiac cycle. What happened when both the atria contract? What happened when the ventricle contract? When semilunar valve open? When atrioventricular valve open and close? So that, I hope that you can remember these notes. The blood vessels that carry blood to and from the heart are labeled A to F. You are seeing this leveling. Draw one arrow on figure 1.1 to show the pathway taken by blood from a vein into the right ventricle. And you can draw this arrow from A, look from A till this ventricle, or from here, there, B, because you know that from the head also, one is superior vena cava, one is inferior vena cava. From B also, you can keep it down. So the arrow, look at my cursor, you can follow the arrow from A to here. So vein to the right ventricle. So from A, you can draw one arrow to the right ventricle. Now, go to the next question. Tell me, identify the letter of the blood vessels that carries blood at highest pressure. And aorta, your answer is correct. Tell me the letter. Which one is indicating aorta in this figure 1.1?
C.
Excellent, C. Now, suggest one causes that valve 1 and 2 close during the heartbeat. Valve 1 and 2. First, you need to understand valve 1 and 2 is semilunar valve, right? And why semilunar valve will contract?
Because the relaxation of ventricles.
Ventricle relax. Okay. So the suggest the cause the valve at 1 and 2 to close during the heartbeat time. What is the reason? When ventricles relax, it increases volume of the ventricles, right? High pressure in aorta and the pulmonary artery, then the ventricle causes semilunar valve to close. Okay, so when the ventricle will contract, that valve will open, right? And the blood will be, from right side, the blood will be, through which passes? Through the pulmonary?
Okay.
Pulmonary artery. And that left side, blood will go through the aorta, right? So that when the blood will be in the aorta and pulmonary artery, higher pressure in aorta and pulmonary artery, then the ventricle causes semilunar valve to relax, closes. Okay, and in that time the ventricles will be in relaxing position. Now they are asking what is the function of the valve 1 and 2. See, any kind of valve.
To ensure blood flows in one way.
They are asking their atrioventricular valve also. Any valve function is to stop Backflow. Yes.
Backflow.
Now, this is again a different question. Figure 1.1 shows that phase of the heartbeat when ventricles are filling with the blood. See, ventricle fills, then ventricle will contract. From the ventricle, blood will go from the left side, blood will go through the outer to the body. From the right side, through the pulmonary artery, it will go to the lung, right? Now, by using this diagram, they are asking, describe and explain how the blood travels from right ventricle to the lung. So this is not too much. Look at this diagram and identify the position of the right ventricle. So right atrium, this one, and down there is right ventricle. When right ventricle contracts, okay, it will increase the blood pressure. So the blood pressure is higher in the ventricle than the arteries, right? So this causes, when the blood pressure will be higher in the ventricle, this creates, this pressure causes the semilunar valve or valve 1 to open. As they're asking us, the question is describe and explain how the blood travels from the right ventricle to the lung. So we will talk about these only. We'll not talk about the left ventricle function. So when the right ventricle contracts, it increases blood pressure. Blood passes in higher, the blood pressure is higher in ventricle than the arteries. This causes semilunar valve or valve 1. Look at the diagram. Valve 1 to open. Then blood flows from right ventricle to pulmonary artery, or you can write D also, you can write C also, same, because this is the branching of the pulmonary artery, or C or D by E. Semilunar valve closes when the blood in pulmonary artery, when the blood will be in the pulmonary artery, semilunar valve will close because ventricle relax and stop and prevent that blood to go backward. Through the pulmonary artery, blood moves towards the lung. Boys, do you have any question? Is it clear? I'll tell the answer again. Look at the diagram and listen. They're asking how the blood is moving from this right ventricle to the lung. So right ventricle, after that semilunar valve, after that pulmonary artery, then the lung, right? So the pathway is easy. You just need to write how it is contraction, relaxation happen. So when the right ventricle, when the blood has come from atrium to the ventricle, the atrioventricular valve has closed, right? Now, when the right ventricle contracts, it increases blood pressure. Blood pressure is higher in ventricle than the arteries. This causes semilunar valve or valve 1 to open. Then blood flows from right ventricle to the pulmonary artery or D/E. Semilunar valve closes when blood in pulmonary artery, because ventricles relax and stop backflow of blood. Through the pulmonary artery, blood moves to the lungs. This is the question. Now, next question. Question number D, total 10 marks of this whole question. D is 2 marks. State the name of the part of the heart, level 3, in the state its role of the mammalian circulation. So what is, look at the diagram and tell me the name of C. What is the name of C? Boys, huh?
Yes.
Yes. What is the name of
Septum.
septum?
Septum. Yeah.
What's
Septum.
C?
What is its function? What does it do?
It separates the
It
oxygenated and
separates
unoxygenated
the
blood.
Separates the
oxygenated blood from the
oxygenated blood from the
oxygen.
allow double circulation.
Yes, both the points are right. It separates oxygenated and deoxygenated blood and allow a double circulation. Thank you, boys. I think that 10C are not joining the class, right? Let me check. See, these here, all the students are from 10B. None of them are from 10C. And this, I want to report it that 10C, they are too much intelligent, right? Too much smart boys, or 10C. That's why now they're not thinking that they should do the class. Anyway, question number 2. Quinoa, right? Senopodium quinoa. This is the scientific name of quinoa. This is a dicotyledonous plant that produces seeds that resemble those of cereals such as rice. This is like one kind of grain, okay, they are giving. So one feature of all dicotyledonous plant that you know, there is nothing to discuss. You can write anything. Broadleaves with branching vein, have petioles, flower pairs multiple with 4, whatever you do. Now, during the seed development, amino acids are converted into storage protein. And that when the amino acid will convert it to storage proteins, and the proteases enzyme will work on it, you know this thing. So protease, that molecules become active when the seed absorb water. And enzyme will not start working if there is no water. That also you know. That's why the seed has to soak in the water. Protease molecule become active when the seed absorb water at the start of germination. Now, the figure they have shown, this figure shows the formation of a storage protein and a protease in developing quinoa seeds, and the action of protease on the storage protein during germination. Now, look at this amino acids in the seeds. Now, this protein will synthesize. Okay, this is not breaking enzyme, right? That Biller enzyme is working like that. So it will break down. That if you have anyone is having connection with 10C boys, please inform that we are going to, because I noticed yesterday only Ifrat joined. First 2 period they have joined. This week I didn't see anyone to bio class. Now that protein synthesis, that this is building enzyme, right? Not the breaker enzyme. So they are making protein from the amino acid. So supply of amino acid in the germinating seed, or they are breaking. Look at the protein, part of the storage protein molecule, and the protease enzyme. This is breaking down process, and finally supply of amino acid in the germinating seed. So here they are breaking down the protein. Now define the term gene. See what is the question diagram and what is the question. During the seed development, the quinoa, some genes are switched on. Okay, that's true for everyone. Some part of your body, some genes are switched on, some genes are switched off. So switching off and switching on, maintaining the characteristics, even though you are having the same characteristic in your whole body. So the length of DNA that codes for a protein that you know. Now describe the differences in the structure between storage protein and the protease shown in figure 2.1. Look at the storage protein. Yeah, look. Shapes. This is the storage protein molecule, and they're asking the protease. Part of protease molecule. So what are you seeing? That storage protein and protease has different sequences of amino acid, right, boys?
Yes.
Different sequence, 2 marks, 2 different points you need to get. So storage protein and protease has different sequences of amino acids. They're composed of different amino acids, right? Both have different shape and both have folded differently. Look, I have told you 3. I told you 3 points. See, storage protein and protease has different sequences of amino acid. They compose different amino acids, and both of them have different shape and they have folded differently. Now, we will go next question. Next question, state the roles of mRNA and ribosome in protein synthesis. That is coordination, chapter. That notes you have. See, mRNA, what does it do during the protein synthesis time?
It carries copy of gene.
Yes,
it determines the specific sequence of amino acid.
I think you people open your marker scheme in front of you instead of the question. Could you please close the marker scheme?
Not me.
Not
because
me,
you're talking language, sentences, everything like marker scheme. Please, boy. You people are smart, I know, but too much smart sometimes, you know, that you are going to catch in the trap. So please close your marker scheme. Don't tell me the marker scheme language. What is in your notes? According to that, tell me the functions, the role of mRNA. What does it do?
It acts like a template and copies the thing.
Good. mRNA acts as a template, or DNA act as a DNA one of the
Oh,
base
sorry.
actors is
The strand, the
forgot. All the
DNA.
notes has got.
Yeah, no, I didn't forget. It's like
Okay, tell the
I
answer.
confusion.
But don't tell me the mark scheme language.
Okay. So
Opening mark scheme and seeing and telling.
one of the strand acts as a template, and then mRNA copies the strand and then goes out of the nucleus and into the ribosome, and then the ribosome.
Good. Actually, the main thing, mRNA copies the DNA-based sequences, then it comes out from the nucleus through the nuclear port, then attached to the ribosome. Okay, so where the ribosome can read out. So this is the thing, and you are all determined the specific sequence of amino acid. This line is not in the notes. This line is in the mark scheme. So it shows that you are seeing the mark scheme and telling the answer. Please, next time, if you are like that, okay, I will stop solving past paper. Ribosome, this, what does it do? This ribosome is the part of the cell or organelle that the side of this cell where the protein synthesis happens. Ribosome can read out the messages which has mRNA brought from the DNA or from the nucleus. Okay, then a ribosome read the messages which has been carried by the mRNA, and according to that messages, write that kind of amino acid has been linked together to assemble the protein. So this is the answer. Now, next question, if you are looking, that researchers investigated the effect of pH on the activity of the protease in quinoseeds. Look at this. State two factors other than pH. What are the other two factors that also that would affect the activity of enzyme?
Temperature and
yes, two factors.
yeah,
pH has given, right? So temperature, of course, high temperature, that low temperature, and optimum temperature. There is if temperature has a great role on enzyme activity, and also
enzymes. Yes,
what
the
type of enzyme is there?
Yes.
So type of enzyme or type of substrate also these are also factors that affect the enzyme activity. So, and sometime that you learn in the notes that surface area, right? Surface area also affected the enzyme activity. The more surface area, the more enzyme that can work well and more product will come. So the surface area and the concentration of the substrate, these are also that affect the enzyme activity. Now, describe the effect of increasing pH on the activity of the protease in quinoa seed. Now, look at this diagram. This is very easy. Could you please tell me the optimum range? Which one is the optimum? That optimum is?
4.
4, right? And
yes.
that more than 5, right? That 5.8 like that is the activity arbitrary units. Now, this curve you need to explain. So, listen to me the way I'm telling. As the pH increases, the activity of protease in quinoa seed also increases at the beginning, right? And
yes.
you can write that it increases gradually, and then optimum pH is 4 because it gives the highest enzyme activity like 0.55 arbitrary unit. From pH 4.3, the enzyme activity decreases, and pH 5, the enzyme activity was 3.5. So it is decreasing, 3.5 arbitrary unit. And at pH 6.5, the activity stops and becomes 0 arbitrary unit. No activity beyond pH 6.5. Understood? So the way they have given you the curve, you need to explain exactly same way. So I'm just telling you again, look at the graph, boys. As the pH increases, the activity of protease in quinoa seed also increases at the beginning. So it is increasing gradually. It's not steeply, boys. Gradually, look, there is a curve. So that the optimum pH is 4 because it gives the highest enzyme activity like 0.55 arbitrary unit. From pH 4.3, the enzyme activity decreases, and at pH 5, the enzyme activity was 3.5 arbitrary unit, and at pH 6.5 enzyme that stop working, and it become the enzyme activity becomes 0 arbitrary unit. No activity beyond 6.5. So this type of that diagram, if it is coming, it will not be a problem for you to solve. Just that look at that graph, and you need to explain starting phase and the optimum phase and the last denaturing phase. So these three, the whole card you will explain with 2-3 data, and according to that you have to give the enzyme activity. Now we will be moving on next question. Explain the activity of protease in at pH 4 and at pH 7. So pH 4, the specific to pH they told us. So pH 4, pH 7.
pH 4 is optimum for it.
Optimum temperature, optimum
And
pH.
for 4
pH.
marks, boys.
What?
For the question contain 4 marks. So total 4 marks. How can you explain? In that time, you need to explain why pH 4 it is high and why pH 7 it is stop. Like,
Because more
At pH 7, the enzyme gets denatured.
yeah, because the active site is no longer complementary to the
Yeah, and the
active site becomes deformed.
enzyme changed the shape.
okay, that's 7. And the opposite thing you have to explain for 4. At pH 4, enzyme fit with the substrate and form enzyme substrate complex because the shape of the site are complementary, and the protease work faster. At pH 4, more effective collision between enzyme and substrate, and that shows more enzyme activity. But that at pH 7, the enzyme denature, active site change the shape so that substrate couldn't fit and do not make the enzyme substrate complex, no effective collision. So as the enzyme denature, no enzyme activity. So to get the 4 marks, you have to explain both the things. So why at 4 it is working? Because the substrate and enzyme fit together, form enzyme substrate complex, because the shape, both of their shapes are complementary, and that protease work faster than more effective collision between enzyme and substrate. But opposite you will write in pH 7. Okay?
Yeah.
Now, next question. See, stem cell, I have found that different questions are coming in stem cells, that 2 marks, 3 marks. But this is the variant. There's a big question about a stem cell. How? Look at this. See, this is the, it shows how the red blood cells has become specialized from the stem cell. Look at the first cell. This is nucleus, mitochondria, and they're like normal cell. It is not specialized like red blood cell. Now, this cell become 2. This stem cell turn into 2 stem cell, and then again turn into 2. See, from 1 stem cell, it become 1, 2, 3, 4 stem cells, right? And which process? By which process? Which cell division, boys?
mitosis.
Mitosis. So this stem cell has been divided into mitosis, and one of that stem cell has specialized into red blood cell. And how this? Look at the question first. This question, it has 2 parts. Use the information and describe how red blood cells are produced. This is one part of the question, and explain how they are adapted to their function. So this question has 2 parts. Now go back to the diagram again. So our first part of this question is how red blood cells are produced. Later I will explain how they are adapted with the function. But adaptation with the function, that is in your notes, chapter 2, red blood cell adaptive structure, adaptation, and their function. This notes you have in chapter 2. But this is, you need to understand by looking the diagram. If you are looking, look, stem cells divide by mitosis division and produce genetically identical cells, right? Only one of the stem cells specialize to produce red blood cells, and other continuing as stem cells. What happened? Look at this picture. Look at the arrow. First, the nucleus of the stem cell bursts off. Look, the nucleus is moving away. First, the nucleus of the stem cell bursts off because there is no nucleus in mature red blood cell. After removing that nucleus, look at this picture. After removing the nucleus, mitochondria has broken down and digested because there is no mitochondria in mature red blood cell. And so this is the way this stem cell turn into red blood cell. So the formation has cleared. Now, again, I'm telling the formation. Listen, stem cell divides by mitosis, division, and form many genetical identical cells. One of the stem cells specializes to produce red blood cell and others continuing to be stem cells. First, the nucleus of the stem cell bursts off because there was no nucleus in mature red blood cell. After removing the nucleus, mitochondria has broken down and digested because there was no mitochondria also in mature red blood cell. Now, second part of this question is explain how they are adapted to their function. See, the loss of nucleus and
loss of nucleus
gives
provides more space for hemoglobin.
To transport more oxygen. And this biconcave shape gives them the large surface area compared to the volume ratio. The shape also helps to pass through the capillary and diffusion of the oxygen. So, boys, this question 6 marks. So you need to write carefully that both the part you have explained properly to get full 6 out of 6. And the liquid part, blood plasma, you know. Next question, stem cells are also found under the outer layer of the skin. I told you that adult stem cell, epidermal layer. So explain why stem cells are found in the skin. Stem cells are found, why? Yes, tell.
Because every second the skin.
Every time we are losing, right?
The cells are being replaced.
Every time we are losing epidermal layer of the skin to replace the cell that has died, rubbed off, or exfoliated, whatever. So that is stem cells are found in the skin to replace that cell that you have been lost every time. Or also that it can heal your wound. 2 points you can give. Next part is easy. State the nutrient that is deficient in the diet. Anemia. What nutrient? Lack of nutrient. One of the lack of nutrient causes anemia. What is the name of that nutrient, boys?
Iron.
Iron. And the symptoms, 2 symptoms of anemia?
Fast breathing.
Dizziness.
Yes. Wait,
Dizziness.
miss, I know.
Yes.
Wait. Like what do you do when you like like that oxygen depth?
What he's telling? Hey, what are you telling? Means
Panting for oxygen.
Heavy breathing. Yes, heavy breathing.
heavy breathing. Shortness of breathing.
For that you heavy breathe, right?
Listen, the main point, when the oxygen supply will be less, your body couldn't supply sufficient amount of energy, right? And lack of energy will make you tired. So why don't you answer this, that fatigue or tired or weakness will be a great symptom of anemic patient. And as they're anemic, their infection, their wound, this will not heal that on time, and they will suffer infrequent infection. So I told you that those who are weak people, they are seasonal, all the diseases they will get. So this is the answer, and this that person couldn't do his that everyday's work, and within that simple reason, that person will be tired. So shortness of breath, dizziness, and that frequent infection, fatigue, tiredness, weakness, these all could be a symptom. Now, describe see, some people have sickle cell anemia. These are the, like common question from selection, that variation chapter, right? Describe the cause of this type of anemia, sickle cell anemia, and you know this thing, right, boys? Can you remember the notes, or you forgot?
Yes, miss, we remember.
You remember, Mahir, or you forgot? Which one you are?
I remember.
Oh, so tell me that sickle cell anemia caused by what is the reason sickle cell anemia
Because one of the
change in base
because
sequence
yeah,
of
change in base
DNA.
sequence.
So
Yes,
change in
and
base sequence of that, how does it happen?
radiation, exposure to radiation.
When the radiation
Yeah,
occurs,
it
what is the
causes
term we are
mutation. It
using?
causes
Mutation.
Good.
Mutation.
Sickle cell anemia caused by mutation, which changes DNA base sequences for hemoglobin. And due to that mutate hemoglobin, right, HBS, RBC become distorted and sickle shape. You know the sickle shape, how does it look, right? So RBC or red blood cell cannot supply sufficient oxygen to the body cells, and it can sometimes block the capillaries. A person, those who have recessive allele like HBS cannot survive and die before puberty. But the carrier parents, those who are heterozygous, they have normal life because this is codominant, right? So it can pass to the mutant allele to their child. And so in this way, people can inherit this disease also. Okay, so suppose if both the parents are heterozygous, then 25% child will be sickle cell anemic patient. So in that cases, but those who are sickle cell recessive allele, they cannot that forward this characteristics because they will die before their puberty. They couldn't complete their reproduction. So this is the answer. Remember, sickle cell anemia caused by mutation which change DNA base sequence for hemoglobin. Due to muted hemoglobin, RBC, that is HBS, right? RBC become distorted and sickle shape. RBC cannot supply sufficient oxygen to the body cell and can block capillaries. A person, those who have recessive allele like HBS cannot survive and die before puberty. But carrier parent or heterozygous parent have normal life and they can pass this mutant allele to their child. So people, in this way, people can inherit this disease also. So next question. What are you seeing in this photograph?
We are seeing.
Oh, thank you. You understood this is a bird.
This is a what is
This is what, Mahir?
my friend?
This is Mahir's friend. I understood. Stuck in the that home. No school, no friends, no conversation. So this picture, it's Mahir's friend's picture. Okay. So Mahir's friend name is Amazona barbadensis. This is Mahir's friend. Okay.
Allah, Barbie.
Yeah, Barba, not Barbie, huh? Barbadensis. So this photograph of a yellow shouldered Amazon. This is the name of the local name, and the scientific name is Amazona barbadensis. This is the one type of parrot actually. This you can get it in Venezuela coast of Caribbean, that area. You will get this kind of the parrot over there. But they're asking which vertebrate group, sir, is it?
Birds.
Birds. And do you know another name is also there, birds? That is Aves, right? Aves. So what is?
Yes.
It has two special features. So which shows that they are birds, which you will not get in other that vertebrates. Tell
wings.
What is that feature?
Wings.
What, miss?
Wings and
Insect
beak.
also have wings, right?
Oh, feathers.
Feathers.
Sorry, feathers
Sorry.
and beaks.
Beak, excellent.
Eyes,
They
nose,
do not have
leg,
teeth
everything.
also. Anyway, Mahir has teeth, lot of teeth. Now, this species is subdivided into several populations on the mainland and the island of Margarita and Bonaire. Scientists believe that yellow-shouldered Amazons rarely travel between these two places. That is the scientist's prediction. Now, this is not the question. This is the descriptional line they are using to make you more confused. Explain what biologists mean when they refer to population of animals such as Amazona barbadensis. You need to define population with this example of this parrot. Now see, this type of question, when they're coming, you will just write the definition of the population. Then you will give the example as Amazona barbadensis. So you are going to start like that. Biologists mean group of individual animals when a group of same species living in the same habitat or same area at the same time. This is called population. Like all the Amazonia barbadensis living in one area is called their population. So for 3 marks, you will write the definition, and definition also will change a little bit because they're asking what does the biologist mean? So you will write biologists mean group of individual animals when a group of same species living in the same habitat at the same time is called population. Like all the Amazonia barbadensis living in one area is called population. And of course, scientific name you have to underline separately and maintain genus and species name capital and small letter. Okay, so boys, I'm running out of time. I'll give you the link again. I have less than 1 minute. Okay, I'm coming.
Okay, miss.
Okay, good morning, everyone. This is our second session. And look at the screen. We are going to start from the question that we have stopped. Look, this question. This is page number 12. Page number 12. This is page 12. Now this question will go down. Page number 12, this question, look at this C. The number of yellow-shouldered Amazon of Margarita Island has decreased to 700 parrots by 1989. So you can understand the organism becoming endangered. The population then increases 1600 parrot by 2009. Part of this increase was due to the release of the captive bred parrot on the island. So they have increasing their number of the parrot artificially in the captive breeding program. This is one of the few successful release programs of parrot. A similar release program in Arizona in 1980 of the different species of the parrot was not successful. So similar they are trying to carry out same program for different species of the parrot in Arizona, but that was not successful. Boys, 2 marks. This is from your common sense. This question answer you will not get from the notes. Suggest why release program for captive bred parrots are not successful. You know the disadvantages of the conservation.
They're in wild. They have to find their own food. So
yes, the parrot that are not able to find the food, or the parrot. First of things, the competition for the species with this wild that environment with the new species, and they are unable to survive by doing this competition in presence of the disease or in presence of the natural situation, how to find out the food. So one point you will write like that: the parrot may die or killed by the predator, and they will not be able to find their food, and they are more susceptible to disease. Now, and one more point, that 2 point you need to write, right? One more point you need to write, that is, look, second point, whenever you are keeping that animal in a captive breeding, you are providing all the things they need to survive, right? So captive animals unable to adjust in the wild environment, and sometime with their own species, another that organism, they couldn't adjust with them. Same, if you are keeping any organism, like if you have a pet, if you're releasing that pet into outside, they couldn't survive because they don't know how to fight with other organisms to get the food. So these type of things, so captivating organisms are unable to adjust or cope with the wild, and cannot adjust with the wild population of the parrot. And sometimes, one more thing, captive breeding, that few number of parrots you are keeping. So within the few species that new species are coming, so which can increase genetic that disorder or low genetic variation. Due to this reason also sometimes that these programs are not successful. So any 2 points you can write that cannot get the food, cannot survive, killed by the predator, more prone to the diseases, more cannot survive by compete with the existing organism in the wild, are captive animals are unable to cope with the wild animals, like that. 2 points are enough to get the 2 marks. Now outline the disadvantage of this captive breeding program. So disadvantages, first of all, that for 3 marks you need to explain this. There is not enough animal to breed in captive breeding program, right? Because less number of, when the organisms number are endangered, you are keeping them in the captive breeding to increase their number artificially. So there is not enough animals to breed in captive breeding programs, and due to inbreeding, see, so small that members between them that breeding process is going on. So breed between a small number of the animals causes less genetic variation, and this increases the risk of genetic disorder or genetic diseases. Animals also less likely to adapt or to adjust or cope with the changes in the environment, like habitat change, or that any kind of disaster. Naturally, there are lots of disasters. They couldn't adjust with this all, and or maybe the environment got some new diseases or got some new species. They couldn't adjust. These are all that one type of disadvantages. And another important disadvantage, that is, this program is very costly. Sometimes if that many government, if they want also, they couldn't run this program for a long time because it's very costly. Now do you know in England that America, in I forgot that name of this Jew, they found one of the tiger became very sick. So they did the test and they found coronavirus positive in the tiger. I don't know that is right or wrong. I read from one article. So
wait, like animals are like,
I heard that animals are not having this, but I read one cat that this cat got it from its owner, and now yesterday I read that tiger got this thing maybe from the person who is giving food from that person.
actually,
Many biologists think that it is better to conserve ecosystem rather than individual species such as parrots. Explain the advantages of conserving ecosystem. Understood? Conserving ecosystem maintains biodiversity and saves many species and prevent extinction. It prevents disruption of food chain and food web. It gives shelter and breeding grounds for many species. Conserving ecosystem also provides service for human, like reduce the natural disaster, greenhouse effect, maintain the balance of carbon cycle, water cycle. It provides food, fuel, drugs, and raw materials for medicine. It also provides genes for the future genetic engineering. And also it can be used for an area for ecotourism and recreation. Understood?
yes.
Okay, there are actually 3 marks. I told you many points. Again, I'm telling you, just listen. Conserving ecosystem, maintain biodiversity, and saves many species and prevent extinction. 1 point. It prevents disruption of food chain and food web and gives shelter and breeding ground for many species. 2nd point. Conserving ecosystem also provides service for human, like reduce natural disaster, greenhouse effect, maintain the balance of different nutrient cycle, provide food, fuel, drugs, and raw materials. 3 points. You'll get 3 marks. But as the 3 marks, they have given more space, so you have to write a bit more. Next question. An investigation should study the effect of fertilizer on grass yield and species diversity in the grassland ecosystem. Some plots within the grassland were treated with fertilizers containing nitrogen, magnesium, and phosphate. The control plot did not have any fertilizer. See, so the plot with fertilizer, they give this amount. In plot with without fertilizer, the control plot give this much amount. Look at the differences between the crop yield. Calculate the differences between the average yield of two plots. So how differences you are going to find? 1733 minus
1009 divided
do
by
it.
1.
Do this 1733 minus 1009 divided 1009 multiply 100.
My calculator is not here.
His answer is 72%.
Yeah, that what will be the without whole number?
72 point. Allah clear kollam kya?
It's not 72 point. Maybe you are that again. You are seeing mark scheme. That's why.
Yes, no.
Again, that's why I'm
72.
asking you. 72% is the whole number. What is the answer?
71.75.
Yes, this boy did the maths. 71.754. So you will take the whole number answer. This is 72%. So maybe it's Muntasir who is saying the mark scheme and telling the answer.
Yes,
Actually,
exactly.
you should give the exam. Your habit has destroyed completely. Explain the average yield increased as a result of adding the fertilizer. So if you look at these two data, you'll be seeing that look at these 2 points, plots with fertilizer and plots without fertilizer. So fertilizer provides different ions, salts, and minerals required by the plants. And plant needs nitrogen ions to make amino acid, protein, DNA, RNA. They use this protein for their growth. They use magnesium to make chlorophyll. More chlorophyll increases the rate of photosynthesis and increase the that average amount of yield. These things you need to explain completely. Again, I'm telling fertilizer provide different ions, salts, and minerals which the plant need to grow and survive. So plant need this, and then after that you will explain separately. Suppose that plant need nitrogen ion to make the amino acid, protein, DNA, RNA. They use this protein for their growth, magnesium to make the chlorophyll, and the more chlorophyll will increase the rate of photosynthesis and also increase the average amount of yield. More photosynthesis, more that amount of food will produce, and more that crop you will get. State the name of the process that occurs when fertilizer washed off. What is the name of the problem?
Eutrophication.
Causes algal bloom.
Yes, yes, eutrophication.
Eutrophication. Good. Next question. Look, again the graphical representation. The researchers counted the number of different plant species in each of the plots in the grassland to determine the species diversity. Counting started at the beginning of the growing season and was repeated every 3 weeks. The average number of the plant species in each type of plot is shown in figure 5.1. Now look at this. Here, the black color, this bar shows the plot without fertilizer, and the light color was showing the plots with fertilizer. And this is this side, this is the time. X-axis is showing time, and this is showing that species diversity. Now, describe the effect of fertilizer on the plant species diversity. This is easy. Just you describe for 3 months. See, fertilizer decreases species diversity. As time increases, the diversity increases, right? Look at the graph. Fertilizer decreases species diversity. As time increases, the diversity increases. Now give data. From 6 weeks, diversity decreases, and 21 weeks, the differences is greater. The average number of plant species for the plots without fertilizer was 15M-2. And the plot with fertilizer was 3M-2. At 21-week plots that with fertilizer, the species diversity increases, and at 24 weeks, it decreases. But with fertilizer, it's not following the trend, and it is increasing. So one is decreasing and increasing, and another one also increasing and decreasing. That process also how increase, decrease, increase. This also you can. My God, Mizan want to come now. Where he was? 3 minutes left, and he came now. Anyway, Next page, that page 16, suggests why some species of the plants survive in grassland and others do not. See that some plants have some adaptation which help them to survive in the grassland. See, some species compete much better than others. They can compete and get the resources like water, space, light, minerals, that ion, and grow faster. So some of the plants have some special adaptation which help them to survive in the grassland, like taller stem, larger leaves, longer roots. This helps some species in grassland to survive well and grow faster. Some species do not compete successfully or that affected by the diseases or the pests and cannot survive grazing by grassland herbivores. So I have told you many things. You don't need to explain these all. Only 3 points you can explain here. Like that some species compete much better than others. They compete and get the resources like water, space, light, mineral ions, and they can grow faster. And some of them have some adaptation like taller stem, larger leaves, longer roots. These all help the species to survive well in grassland. Okay, so these are the answer of this question. Now, that next question, actually question number 6 is big. And what I feel I will not be able to finish today, no problem. This question I will explain tomorrow. Tomorrow I will explain one paper 6, and along with the paper 6, I will come to this question number 6 also. Boys, you know the meningitis, right? Transmissible disease. This one form of the disease caused by bacterium. Neisseria meningitidis. This is the scientific name of the bacterium. Neisseria meningitidis. So Neisseria meningitidis. This is not a question. They're asking you, what is transmissible disease? This is the definition from the notes. Diseases which caused by a pathogen and can pass from one organism to another. Okay, this is called transmissible disease. So tell me, coronavirus, is it a transmissible disease?
Yes,
Good.
it can transfer.
Yeah, so it can transfer from one person to another person. So this is transmissible disease. Now we will be doing this. You can do. State what is meant by a nerve impulse. This also from the coordination chapter. This is the definition. Nerve impulse, electrical signal which passes along a nerve cell or the neuron in one direction. This is called nerve impulse. So boys, from question number 6, I will explain again tomorrow, and I will give you the tomorrow we will solve one paper 6. That's 17. Any variant, I will send the link. Okay. Thank you for.
Thank you, miss.
Thank you. Assalamu alaikum.
Thank you.
Yes. Bye. Bye, Mahir.
Bye.
Bye, everyone.
Ma salama, miss.
Allah Hafiz, miss.
Ma salama.